Hello and welcome back. So in the last web lecture, we looked at part one of photosynthesis. We looked at the photo part, and we saw how photons of light with particular wavelengths are captured and absorbed by the pigments within plants, the chlorophyll pigments and other accessory pigments. We saw how those photons raise the energy of electrons in those pigments and how that potential energy in the electrons was transferred from pigment to pigment into photosystem number 2, and how that activated electron, that high energy electron, ended up being transferred between the two photosystems and finally to NADPH. So this is where we left off our electrons in the light reactions. We ended up taking electrons from water by splitting the water molecule, releasing oxygen, molecular oxygen in the process. So remember, the production of oxygen is not a benefit to the plant in any way. It's basically a byproduct or a waste product of those light reactions. And then stripping those electrons from the hydrogen, leaving hydrogen ions and electrons being transferred to NADP plus to form NADPH, which is then going to be fed into the Calvin cycle. Also, along the course of transferring this electron through an electron transport chain, we ended up synthesizing some ATP, which is also going to be used in this next part of the process, the synthesis part. And this is where we're going to actually produce the sugars using up carbon dioxide, fixing carbon dioxide, and taking those carbons to build these sugars that can be used for energy. They can be used as carbon backbones to synthesize other molecules. This is where basically all of that biosynthesis happens. So first, let's take a close look at the Calvin cycle. So the Calvin cycle uses the chemical energy of ATP and NADPH to reduce carbon dioxide to sugar. So just the reverse of what we saw in cellular respiration. So the Calvin cycle, like the citric acid cycle, regenerates its starting material after molecules enter and leave the cycle. So it's going to be somewhat similar to the citric acid cycle in that way. The Calvin cycle is anabolic as compared to the citric acid cycle, which is catabolic. It's going to build up that sugar from smaller molecules by using the energy in ATP to run that endergonic reaction, and also using the reducing power of electrons to raise the energy level of those carbon molecules, and those high energy electrons are going to be brought to the Calvin cycle by NADPH. So carbon enters the cycle as carbon dioxide, and it's going to leave as a sugar named glyceraldehyde 3-phosphate, G3P. If this name sounds familiar, it's because you've seen this molecule before when we were talking about glycolysis. This is the first product that's formed when that 6-carbon sugar derivative of glucose is split into two 3-carbon sugars. So same intermediate there, the glyceraldehyde 3-phosphate, we can just call it G3P. So for the net synthesis of one G3P, the cycle must take place 3 times, fixing 3 molecules of carbon dioxide, one at a time as it goes through the cycle. So a single turn of the Calvin cycle is a 3-step process. It has 3 phases. The first phase is fixation of carbon dioxide. So basically the carbon in that carbon dioxide is going to react with a 5-carbon compound called ribulose bisphosphate. We'll just call it RUBP. That 5-carbon compound, that 5-carbon RUBP, is going to be transformed into a 6-carbon molecule. The 6-carbon molecule is going to have 2 phosphate groups attached to it. It's going to be highly unstable. So it's almost immediately going to split into 2 3-carbon molecules of a molecule called 3-PGA. Don't worry too much about that name. The second phase is reduction. So this is where those NADPH molecules come into play. So the 3-PGA are phosphorylated by ATP and reduced by NADPH, and this is going to produce the glyceraldehyde 3-phosphate, the G3P. Some of that G3P is going to be used to make sugars, so it's going to be kicked out of the Calvin cycle and used to synthesize glucose if two of them are combined together to make that 6-carbon glucose, which can then be put together into more complicated sugars. The next phase is regeneration. We need to get back to that RUBP molecule that we started with. So the remaining G3P that's staying in the cycle is going to be used in reactions that use ATP to regenerate RUBP. So let's take a look at the details here. Here we have the end of the last cycle ending with RUBP. We've got 2 phosphate groups attached to this 5-carbon sugar. We're going to add 1 carbon dioxide at a time. But the diagram you're going to see is a summary of 3 turns of the cycle. The reason for this is that each individual turn of the cycle is incredibly complicated because we're adding 1 carbon dioxide at a time. All of these molecules aren't easily divisible by 3 like what you will see here. Things get shuffled and reshuffled in very complicated ways that you don't need to worry about. So you can think of this most easily, think of the first 2 cycles as something very different happening from what you're going to see here, and no sugar comes off in those first 2 turns of the cycle. You can think of this as what happens on that 3rd turn of the cycle where the 3rd carbon dioxide is added. But we're going to visualize this as all 3 carbon dioxides being added at the same time for the sake of being able to understand it clearly. So 3 RUBP molecules. This reaction is going to be catalyzed by an enzyme called Rubisco. So normally I've told you not to worry about remembering the names of the enzymes involved in these reactions. This is going to be the exception because this is going to play an important part later on in this web lecture. So make sure you have made a note of this name Rubisco. This is the enzyme that catalyzes the fixation of carbon dioxide onto this 5-carbon RUBP sugar. So we've got 3 RUBP coming and joining with 3 CO2s in the 3rd turn of the cycle. The last carbon dioxide is being added. So we have now 3 6-carbon sugars with 2 phosphates attached. Remember, those phosphates are going to be unstable attached to the same molecule. They're going to repel each other. So this 6-carbon sugar is going to quickly split into 2 3-carbon sugars. So now we've got a total of 6 3-carbon sugars, each with 1 phosphate group attached. And this is the carbon fixation. Now that carbon dioxide, the carbons from the carbon dioxide have been incorporated into these larger organic molecules. The next phase is reduction. So in this first step, ATP is going to be used to phosphorylate all of these molecules and add another phosphate group to this 3-carbon sugar. Then these 3 carbon sugars are going to be reduced by NADPH. It's going to donate that electron, and also these 3-carbon sugars are going to have one of those phosphate groups removed back down to a 3-carbon chain with a single phosphate group, and this is that molecule G3P that we've been talking about. So we're using 1 ATP per one of these 3-carbon sugars, 1 NADPH for each of these 3-carbon sugars, and we're going to generate 6 phosphate groups, 1 for each of these, when this phosphate group is removed. So now we've got 6 molecules of G3P for every 3 turns of the cycle. So remember, we're summarizing this as being sort of the 3rd turn of the cycle, and so we're going to release one of these G3Ps as output, and this is going to go on to produce glucose and other organic compounds. The other 5 are going to stay in the cycle. So we have 5 G3P now. Those carbons are going to be reorganized in a very complicated way using ATP, so that these 5 3-carbon sugars are going to be rearranged into 3 5-carbon RUBP sugars, each with 2 phosphate groups, regenerating that original starting molecule. So in summary, for every 3 turns of the cycle, we're going to be using 9 ATP and 6 NADPH, and all of that is going to be provided by the light-dependent reactions. Here's another little summary slide that might help you visualize this. So remember that the Calvin cycle, All three phases are going to be taking place in the stroma of the chloroplast. Remember, at the end of the light reactions, all the NADPH and all the ATP was being dumped right into the stroma. That's where all of this takes place. In the fixation phase, we're going to have 3 RUBP plus 3 carbon dioxide, giving us 6 of those 3 PGA molecules, that intermediate, that's then going to be reduced. So the 6, 3 PGA plus 6 molecules of ATP coming from the light reactions plus 6 NADPH also coming from the light reactions are going to yield 5 G3P that's going to go on to step 3 and 1 G3P that's going to be output from the cycle. Then those 5 3-carbon sugars are going to be reassembled into 3 5-carbon sugars in the form of RUBP, and that's going to continue the cycle, accepting those carbons from carbon dioxide in the next turn of the cycle. And then just another cyclic representation of this, not much different from the one we just looked at. So now that we have these G3P molecules, what's going to happen to those? How is this going to help us in the life of the plants? So the G3P molecules that are produced by the Calvin cycle are often used to make glucose and fructose by the process called gluconeogenesis. This literally means the making of new sugars. Genesis refers to an origin or beginning. Neo means new, and gluco obviously refers to glucose, so new glucose synthesis. And then these glucose and fructose can be combined to form sucrose. So when sucrose is very abundant, then the plant is going to start to store it up in the form of starch. So the glucose is going to be polymerized to form starch to store for later use. And then the starch is going to be broken down into glucose at night. Why at night? Because that's when photosynthesis isn't happening, and that's when the plant is going to use those sugar stores, because it's not producing any new sugar when it doesn't have any light to use as fuel. This is sort of the standard pathway. We call it the C3 pathway because it involves the production of a 3-carbon sugar. So we call this the C3 pathway. But there are other alternative mechanisms of carbon fixation that have evolved particularly in hot, arid climates. So this is a specific adaptation to help plants survive in hot, dry areas. So dehydration is going to be a very big problem for these plants, and this is sometimes going to require trade-offs with other metabolic processes, especially photosynthesis. Let's see why. Plants are basically covered with this waxy coating that's going to prevent excessive evaporation of water. So evaporation of water from the leaves plays a very important part in the plant's physiology, but we want to make sure that it doesn't lose so much water that it's going to dehydrate. And so this waxy coating prevents excessive evaporation, but it also limits the transport of the gases that it needs to exchange, carbon dioxide and oxygen. So plants have little pores that are called stomata that allow gas exchange. And here's a micrograph image of some of these little pores. The CO2 is going to come in through here, the oxygen is going to leave through here. They're going to be surrounded by these 2 guard cells around that can actually change shape to open and close these little pores, these little stomata. So these are under control of the plant depending on the water conditions at the time. So basically they're going to open these stomata to allow carbon dioxide to diffuse into the leaf and oxygen to diffuse out. We see that diagrammed here. Here's one of those little guard cells cut in half as we see this cutaway. Carbon dioxide coming in, going to the chloroplasts, oxygen, the waste product, coming out. Because these chloroplasts are using up carbon dioxide in the Calvin cycle and removing it, there's a constant strong concentration gradient favoring the entry of carbon dioxide that's going to keep the carbon dioxide coming in and obviously not coming out because it's being used up. So stomata are usually open during the day when they're actively photosynthesizing and closed at night because they don't really need to do this gas exchange. They want to conserve the water at night when they don't need to have it open to exchange the gases. So when conditions are hot and dry, that means they're going to be losing a lot of water through these stomata. This is also going to be the way that water evaporates out. So stomata are going to have to close to prevent the evaporation of too much water when it's very hot and dry. This creates a problem, right? It's going to be mostly hot and dry during the hottest part of the day, which is when they're getting the most light to perform photosynthesis. So this is a problem. This is when this phenomenon called photorespiration starts to happen. The carbon dioxide fixing enzyme, remember, is this enzyme called rubisco. And rubisco is found in all photosynthetic organisms that use the Calvin cycle to fix carbon, and it's also thought to be the most abundant enzyme on Earth. Rubisco is a very slow enzyme. It doesn't catalyze these reactions very quickly, and it's also fairly inefficient because not only will it bind CO2, but it can also connect oxygen to RUBP. When oxygen concentrations are high, because those stomata are closed and blocking the oxygen in, and remember the oxygen is being generated constantly by the light reactions, then there's a problem. Then it's going to slow the rate of carbon dioxide reduction. So we're not getting the carbon dioxide in because the stomata are closed. We're not releasing the oxygen out because the stomata are closed. There's going to be more oxygen there than carbon dioxide. This is going to really mess up the Calvin cycle. Photorespiration is going to end up consuming oxygen because it's binding it to those RUBP sugars, and it's going to consume organic fuel because it's going to break down those molecules into waste products because they're not the sugar that can be used to synthesize other things. They're just going to be broken down and discarded. And it's going to release CO2 without producing any ATP or sugar. So we're calling it respiration because it consumes oxygen and organic fuels and gives off CO2, similar to cellular respiration, but it's going to consume these resources without producing any form of energy the way this process would in cellular respiration. So here's a summary of what happens here when there's a reaction with carbon dioxide during photosynthesis. Usually RUBP combines with carbon dioxide using rubisco to ultimately form 2,3-phosphoglycerates, which are going to be used in the Calvin cycle. But when RUBP is connected to oxygen instead of carbon dioxide using rubisco, we get a completely different sugar that's also used in the Calvin cycle, but when it's processed, carbon dioxide is released and ATP is used. Okay, so we are using oxygen, we're using that organic molecule, releasing CO2, but we're using up ATP instead of producing it. So this is a very bad thing for the plant. You might wonder how evolution could have come up with something that is so clearly maladaptive. And so it's possible that photorespiration might be just an evolutionary relic left over from a time when the atmosphere contained far less oxygen and a lot more carbon dioxide, and this would not really be a problem for rubisco to also bind oxygen because there just wasn't that much molecular oxygen around. Most of what was around would have been carbon dioxide, so there would have been no selective pressure on plants to use something other than rubisco because that tendency that it has to bind oxygen would not have been important in terms of survival. But now, in many plants, photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the Calvin cycle. This can be a very big drain on the energy in these C3 plants that are in hot, dry areas. So some plants have evolved mechanisms to deal with this problem of photorespiration. And one group of plants are called C4 plants. So the C4 strategy appears to have evolved many times independently in different groups of plants, and all of them have resulted in a very similar structure and function. So C4 plants minimize the cost of photorespiration by incorporating carbon dioxide into 4 carbon compounds before they enter the Calvin cycle, before they encounter rubisco. So there are 2 distinct types of cells in the leaves of these C4 plants. The first type are called bundle sheath cells, and so they kind of bundle and form a sheath around the vascular tissue, these veins that run through the leaves, and they're going to be tightly packed around them. And then the other type of cell you're already familiar with, these are the mesophyll cells that are going to be loosely packed between the bundle sheath and the surface, always very close to the bundle sheath. They're never going to be more than 2 or 3 cells away from this bundle sheath, and we'll see why in a moment. So let's look at this mechanism for increasing the carbon dioxide concentration when the stomata are closed during hot, dry circumstances. So sugar production in C4 plants occurs in a 3-step process. The first step is the production of the 4-carbon intermediate that we talked about, and that's going to be catalyzed by the enzyme PEP carboxylase, and this is going to happen in the mesophyll cells. So here comes the carbon dioxide. Before it even encounters a rubisco, it's going to find this PEP carboxylase in the mesophyll cell and get processed into a 4-carbon sugar. So the PEP carboxylase has a higher affinity for carbon dioxide than rubisco does. That's why it's going to grab it first. And it can fix carbon dioxide even when carbon dioxide concentrations are very low. It also has essentially no affinity whatsoever for oxygen. So this is a really nice enzyme to be able to use for this purpose. And then these 4-carbon compounds are going to be exported to those bundle sheath cells through plasmodesmata. Okay, so we've got our 4-carbon sugar. If this intermediate oxaloacetate looks familiar to you, that's because it should be. This is that 4-carbon sugar that is regenerated during the citric acid cycle. The 4-carbon sugar, when it gets to the bundle sheath cells, is going to release that carbon dioxide back out, converting the 4-carbon sugar into the 3-carbon sugar pyruvate, which again should be familiar to you. This is the end product of glycolysis. The carbon dioxide then is going to find a rubisco. There's going to be very little to no oxygen here in these bundle sheath cells, so it's going to be a high concentration of carbon dioxide, low concentration of oxygen, and so the Calvin cycle is mostly going to be processing carbon dioxide rather than oxygen. The pyruvate gets exported back again through these plasmodesmata, these gaps in the cell walls, back into the mesophyll cells to continue to do this. So an ATP is used in converting pyruvate back into PEP, which is going to be what actually bonds to that carbon dioxide. So this is an energy using process, and we can think of this as the cost of concentrating carbon dioxide within these bundle sheath cells. The sugars are then transported directly to the vascular tissue to be exported out to the rest of the plant. So that proximity of the bundle sheath cells to the vascular tissue is important and significant. So here's another image of that process. So again, you can see not only what happens, but where it happens. Here we have the leaf surface, and these mesophyll cells close to the leaf surface, they're going to be doing the light reactions and also taking in the carbon dioxide from the surface. Here are those bundle sheath cells surrounding the vascular tissue, the vein going through the leaf, and then a close-up through that junction between the mesophyll cells here on top and the bundle sheath cells below. We've got carbon dioxide coming in, encountering PEP carboxylase, which has a high affinity for it, no affinity for oxygen, being converted to a 4-carbon compound, going through the plasmodesmata into the bundle sheath cells, releasing that carbon dioxide, which is going to enter the Calvin cycle, and release those sugars into the vascular tissue. The 3-carbon compound that's left after the carbon dioxide is released is going to be recycled back into the mesophyll and processed back into PEP to repeat this cycle. So some of the common plants you may know about that do use the C4 pathway are sugarcane, corn, and millet. Another strategy that has evolved in many groups of plants to deal with this problem of photorespiration is crassulacean acid metabolism. We're just going to call this CAM. So this is the CAM pathway. So CAM plants include such species as cacti and other succulents, also things like pineapples. And these are also going to use the strategy of attaching carbon dioxide to a 3-carbon compound to form a 4-carbon compound. And again, this is also, just like we saw in the C4 pathway, it's going to occur in addition to carbon fixation to RUBP by rubisco, that C3 pathway. So it's going to use both. So CAM plants are going to open their stomata at night. So these are plants that live permanently full-time in hot, dry areas. They open their stomata just at night, incorporating carbon dioxide into these organic acids, these 4-carbon sugars that are going to be stored in the vacuoles until morning. So the stomata close during the day when it's very hot and dry and they have the biggest risk of losing lots of water. And at that point, when the stomata close, now the photosystems are open for business, the light-dependent reactions are running, and at that point, the carbon dioxide is released from those organic acids and used in the Calvin cycle. So remember that things like ATP and NADPH are not very stable. They need to be used quickly once they're made. So this is a way of just storing up that fixed carbon and releasing it from within the cell at a time when that ATP and NADPH is available to fuel the Calvin cycle. So let's do a quick comparison between the C4 and CAM pathways. So the CAM pathway is very similar to the C4 pathway. They both use a strategy of incorporating CO2 into organic intermediates before it enters the Calvin cycle. So the C4 pathway has these two steps structurally separated. So in separate cells, but happening at the same time. Okay, so again, we've got the creation of the intermediate happening in the mesophyll cells, the release of the carbon in the bundle sheath cells that are going to go ahead and go through the Calvin cycle. In the CAM pathway, these are all happening in the same cells, but they're separated in time. So the carbon is going to be fixed at night and stored in vacuoles until the morning. In the morning, that carbon dioxide is going to be released from those 4-carbon intermediates and go ahead and go through the Calvin cycle at a time when the stomata are closed, but light still gets in to run those light-dependent reactions. So to wrap up, we're just going to do kind of a general review of the importance of photosynthesis. We know this is a very important process to all life on Earth. This is basically where all the fuel for the entire biosphere comes from. And so the energy entering those chloroplasts as sunlight gets stored as chemical energy in organic compounds. The sugar made in the chloroplasts are going to supply both chemical energy and carbon skeletons, so both the energy and the raw materials to synthesize the other organic molecules of the cells. So when plants are very productive and they have excess sugar, they're going to store that sugar as starch in the chloroplasts and also in other structures such as roots, tubers, seeds, and fruits, the kinds of things that we like to eat with all of that stored energy in them. And of course, organisms eat all parts of plants, right? They eat the leaves, they eat the stems, they eat the fruits, and so this is basically the driving force, the source of all of the chemical energy in the biosphere. So now if we think back to this list of things that all living things do, we've been spending the last unit of the course thinking about this second one on the list, this idea of gathering, transforming, and using energy from the environment. We've talked about cellular respiration and photosynthesis. And so at this point, we're going to shift gears and we're going to start to think about cell signaling. So this is going to come into this third category, transmission of information through space and time. In this case, we're going to be thinking primarily about space in the form of cell signaling to coordinate the physiology of that cell. So this is going to also affect two other of these categories. So this is going to be very important to maintaining homeostasis, and this is also going to play a big role in responding to the environment.